Hello and welcome to today's webinar. We'll be introducing the Target EcoTox Challenge. Target stands for Technology Advancing Rapid Gene Expression-Based Testing. This challenge is sponsored by the U.S. Environmental Protection Agency, along with partners from Syngenta, Dow, the European Commission, the U.S. Army Corps of Engineers, and Environment and Climate Change Canada, all organizations that are interested in seeing the development of technologies to support high-throughput testing using transcriptomics-based readouts for 4 different species of ecological interest. What I'd like to do today is provide a brief introduction to federal prize competitions and challenges, give some background and objectives relative to the Target EcoTox Challenge and how it fits in with EPA's broader approach to chemical safety assessment, then give you some information on how to participate in the challenge and what to expect if you participate, and then finally, as time allows, address any questions that you might have. This challenge can be found at challenge.gov. In general, challenges and prizes represent tools that EPA and other federal agencies can use to find solutions to the problems that the government faces by tapping into the ingenuity and creativity of crowds. EPA basically outlines the specifications and criteria for a problem. The public can then submit ideas, proposals, technologies, whatever is needed to provide successful solutions to those problems. And since 2010, the U.S. government has run nearly 1,000 challenges and offered well over $250 million in cash prizes for the best ideas. This particular challenge relates to a particular objective in EPA's strategic plan, and that is ensuring the safety of chemicals in the marketplace. Challenge itself is being conducted under the Chemical Safety for Sustainability program, and the basic problem that this program is intended to address is one of tens of thousands of chemicals that are currently in use and hundreds more that are introduced into the market every year. Only a small fraction of those have been thoroughly evaluated for potential risks to human health and the environment. The traditional approach to evaluating the safety of chemicals has been the use of animal-based toxicity testing, specifically where an animal model is exposed to the chemical of interest at various doses. And then the endpoints that are examined are the actual adverse effects themselves. So looking at potential effects on survival, growth, reproduction, development of tumors, other types of endpoints or disease that we may be concerned about from a human health or environmental perspective. And while this approach can be effective, it's also very costly, time-consuming, and inefficient to implement. So recognizing the need to provide this information for more chemicals, the need to provide greater breadth and depth of information in order to make good chemical safety decisions, the U.S. National Research Council in 2012 proposed a vision and strategy for toxicity testing in the 21st century that advocated that we transform toxicity testing from a system based on animal testing to one founded primarily on in vitro methods that uses suites of predictive high-throughput assays that assess critical mechanistic endpoints that are involved in the initiation or the induction of toxic effects rather than observing the final adverse toxic effects themselves in organisms. Based on this proposed vision and strategy, EPA and partners implemented initial high-throughput screening programs, including EPA's ToxCast program, which to date has screened well over 3,000 chemicals in about 600 different pathway-based assays, looking at everything from whether chemicals interact with and inhibit enzyme activities, bind to various receptors, activate transcription factors, alter cell morphology, even looking at some small animal models like zebrafish embryos and impacts of these chemicals on those. There's also been the Tox 21 consortium, which multiple federal agencies are involved with, which to date have evaluated over 10,000 chemicals using roughly 25 different assays. So a smaller set of assays, but again, providing extensive chemical coverage. Basically, both these programs have showed that this approach of using suites of high throughput assays can provide breadth of chemical safety information for a large number of chemicals at a reasonable price and in a short amount of time. However, there are still limitations to these approaches, one of the most notable being the potential inadequate coverage of biological targets and pathways. And so while ToxCast incorporates over 600 different pathway-based assays, when we put that into the context of gene coverage of the human genome, for example, it's just a tiny, small fraction covering about 300 genes of the entire human genome. And so the question was, how can we make sure that we have adequate coverage of these pathways? So the proposed approach that was laid out in a 2019 paper by Thomas et al., laying out the next generation blueprint for computational toxicology at the US EPA, basically introduced high throughput transcriptomics using human cell lines and probing the entire human genome, expressed transcriptome, as a first tier in a testing and assessment strategy. Chemicals would be exposed to human cell lines, then the entire human transcriptome would be probed. The data that result would be used to both calculate the concentrations at which we start to see a marked change in the expression of human genes, and also pathway analysis would be conducted in order to attempt to infer the modes of action, the pathways perturbed by these chemicals, and potential modes of action through which these chemicals might cause toxicity. Where mode of action can be identified, that information would then be used to inform additional assays, whether they be in vitro or shorter term in vivo assays that could help confirm the potential activities and also reduce some of the uncertainties in the chemical safety assessment. And so this was the strategy proposed as a way to increase that pathway coverage integrated into an overall hypothesis-driven testing workflow. Of course, the elephant in the room when it comes to addressing pathway coverage is that humans are, of course, just one of the species that EPA is charged with protecting. EPA's mission is to protect both human health and the environment, and there are many other species besides humans. While genes covered and incorporated as part of the human genome many of those genes are well conserved across the animal kingdom, there are certainly differences in genes, pathways, and physiology among some of these organisms, and there are pathways that are not represented in humans that are potentially relevant to critical biological functions of these other classes of organisms. And so how does EPA and other partners go about evaluating the safety of chemicals to these other classes of organisms? So the proposed solution is the development of high throughput transcriptomics-based assays, initially with 4 common aquatic ecotoxicity test organisms. These are organisms that are already widely used for ecological hazard characterization. So for example, in the classification and labeling of chemicals relative to their environmental hazards, focusing largely on aquatic organisms because that's largely where the largest or the greatest exposure potential typically occurs in the environment. The organisms selected would represent 3 key trophic levels. So algae or aquatic plants is an example of primary producers. Crustaceans or sediment-dwelling insects as examples of primary consumers, and then an aquatic vertebrate model of fish as a secondary consumer. By incorporating transcriptomics-based assays with this diversity of different organisms, the idea is to capture some of those key pathways that are not conserved with humans. For example, photosynthetic pathways in plants, pathways associated with invertebrate endocrine systems that don't have analogs in humans or other mammals, and pathways such as osmoregulation, again, unique to some of these other classes of organisms. In order to help make this solution a reality, EPA is already in the process of adapting existing test methods for these 4 species. Test guidelines for conducting these tests in aquaria and beakers in lower throughput systems already exist. EPA is adapting those test methods to a high throughput format, including exposure in a 96 well or even more dense formats, including automation of the dosing and sample processing to vastly increase the throughput of these assays, the number of concentrations and the amount of replication that can be achieved. At the same time, EPA is developing the automated and standardized analysis pipelines so that when transcriptomics data are available, they can be subjected to concentration response modeling in order to estimate benchmark doses or points of departure, basically concentrations at which we start to see a significant biological effect of the chemicals, and then also automated approaches to pathway analysis that can be used to then aid the inference of potential chemical modes of action. The real missing piece right now in this overall strategy of developing high throughput transcriptomics based assays is the detection and analysis technology. So we expect that with these assays we'll be generating potentially tens of thousands of samples per year. We need detection and analysis technologies that are commercially available, that are low cost, but yet provide high quality data and maximize the coverage of the transcriptome of these organisms. And that's really needed to address the potential demand that this kind of high throughput testing system will generate. While there are sequencing technologies available already that can do this for these types of organisms, for using RNA-seq, for example, right now the cost is too high to really make this feasible from high throughput screening potential for these species. And so the solvers are then challenged to come up with appropriate technologies, platforms, methods, and approaches that can help reduce that overall cost while still providing quality of data that's needed to generate concentration response estimates and pathway-based information that can help inform chemical safety decision making. So that's the basic goal of the Target EcoTox Challenge, is to fill that gap. So EPA is looking for innovators who can help usher in the next generation of technologies advancing rapid gene expression based testing. Agency is offering a $300,000 prize to the company, organization, or team that can provide high quality, low cost technologies and platforms for evaluating global gene expression in samples using either RNA or tissue homogenates from these 4 commonly used aquatic toxicity test organisms: the fathead minnow, a fish, Daphnia magna as a representative crustacean, Chironomus dilutus as a sediment-dwelling insect model, and Rhaphidoceles subcapitata as a green algae. So we're encouraging you, if you think you have a winning technology or can adapt your technology for this purpose, we encourage you to apply. So if you are to participate in the EcoTox Challenge, I'd like to go through how you go about participating and what you can expect if you were to participate. There's 4 basic steps I'm going to go through. First, how to register and why to register for the Target EcoTox Challenge. then proceeding to the development of your technology platform and the associated informatics for the 4 species of interest, analysis of reference samples, and then submission of data for those reference samples along with a technology description to actually be evaluated for potentially receiving the award if you have the winning solution. So first step is to register for the competition. The registration form is available at the website shown here. The deadline for registration is March 16th, 2020. In order to be eligible for the award and to compete in the competition, you must register by that date. Registration will make sure that you receive all the relevant communications related to the challenge, that you're provided with a set of samples from these 4 species that you can use in your process of method development and development testing of your platform. And then finally, it will also make sure that you receive a set of reference samples, and analysis of these samples will basically be used to evaluate the performance of your platform compared to the other solvers, and ultimately will be used to determine which is the winning technology. There's also opportunity on the registration form to indicate whether you might have part of the solution but are seeking collaborators. If you're seeking collaborators, just click on these radio buttons. If you indicate that your contact information can be shared with other potential collaborators, we will provide you with email alerts that let you know when there are other new solvers that are also seeking collaborators. We'll provide an updated list of the contact information for any of those individuals or groups seeking collaborators. And then the rest is up to you to reach out to those folks to talk about how you might be able to complement one another, decide whether you want to enter the competition together as a team, and then if you were to win, how you would distribute the award. In terms of who is actually eligible to compete, basically individuals or teams from private companies, academic institutions, non-governmental organization, or independent research and technology institutes are eligible to compete. The competition is open to both U.S. and foreign citizens or organizations. The folks that aren't eligible are any U.S. or foreign government organizations, so government employees should not apply. Likewise, any of the individuals that were involved in the development of the challenge criteria, the award selection criteria, reference sample generation, any other aspects that might represent a conflict of interest are not eligible to compete in the challenge. When you register for the challenge, you agree to the terms and conditions. Most notably, it's important to recognize that this challenge is not a grant. There are no resources awarded ahead of time or leading up to your development. Only the award winner will receive the $300,000 cash award. Solvers will not be compensated for the resources or time they invest in attempting to address the challenge. Only the winning solution will receive the cash award, and only the top-ranked solution will receive the award. We will not split the award among multiple organizations with viable technologies. There will be one winner, and it is up to the solver and their team to decide how those resources should be distributed among their team members. With that in mind, since there are no resources provided up front for the work, the solvers reserve the right to remove themselves from the competition at any time and are under no obligation to deliver results. You can register for competition, start to develop your platform, decide your technology isn't quite where you want it to be, or you can't hit the price point, whatever it might be. You can withdraw at any time. Likewise, if you don't submit your results and technology description template by the submission deadline, you will automatically be removed from the competition. Notably, solvers, any teams that participate in the competition retain the rights to all their intellectual property. So the details regarding your design, the details of your technology. Some of those details may be disclosed to the sponsors, but we will make sure not to disclose that intellectual property without permission. And so basic message is we will take every step to protect your intellectual property. There's no need to transfer the rights to your intellectual property as part of the challenge. At the same time, the sponsors, so EPA and our partners, retain the right to provide a transparent reporting of how the winning solution was selected, including disclosing the reference sample data, the performance criteria, other evaluation criteria that were used to make the decision. And sponsors also retain the right to publish, present, or otherwise publicize the results of the challenge competition as long as it does not involve disclosure of the intellectual property of the solvers. Once you've registered, everyone involved in the competition can develop their platforms. The goal is to develop a platform for all 4 species. Is not sufficient just to demonstrate the ability of your platform to function for one of the 4 species. In order to get maximum points, you need to develop a viable platform for all 4 species, and points in the evaluation will be lost for any species for which platform is not developed. The technology itself must be capable of precisely quantifying the expression of individual genes, providing complete or near complete coverage of the expressed genome. That might be through direct measurement of all those expressed genes or use of approaches like sentinel gene sets, whose expression can be that can be used to confidently infer the response of the rest of the genome. If you choose to take that sentinel gene approach, it's incumbent on the solver to demonstrate that that gene set is indeed representative of the response of the entire transcriptome and provide the means through which to interpret the data in that context. The technology should also facilitate gene-specific annotation to the extent that the genes can be annotated, so that changes in the gene expression profiles can be linked to particular pathways and used to infer modes of action. Technology must also be capable of performing analyses with limited sample masses, because these tests are going to be done in 96 well plate format with individual organisms in each well. Will be fairly limited in terms of the RNA available for each sample. Ideally, the platform should be able to perform with 0.5 microgram of total RNA or less, and platforms that can demonstrate the capability to perform with even less RNA, that will be certainly taken into consideration. Platform should generate data in a format that's compatible with standardized and automated quality assurance and data analysis workflows. We'll talk a little bit more about the data reporting format for the challenge itself. And then the platform needs to be economically and commercially viable for high throughput screening applications. Against it's expected that once these high throughput screening programs are implemented, we'll be generating tens of thousands of samples per year that will need analysis. There has to be a clear demonstration that there's a plan for how the technology can meet the demands of a high throughput screening program in order for that solution to be viable. To aid with the development of these platforms, again, assuming that not all groups that participate in the challenge may have easy access to these 4 species, we as the sponsors will provide a pool of RNA for each of the species to support your method development. We can also provide things like crude homogenates if you want to be able to demonstrate capability of your technology to provide cost savings by, for example, just analyzing crude homogenates rather than purified RNA, or if you want to show capabilities to handle a different type of sample. Once your technology is developed, each solver will then analyze a set of reference samples. Those reference samples will be provided to you by September 18th of 2020. Sponsors will be providing 36 reference RNA samples, basically 9 samples per species. Solvers will be asked to analyze each of those samples using their technology and submit their results along with a technology description document. The evaluation of this data will be used to judge which will be the winning award. These reference samples will be pooled such that all participants in the challenge will receive basically aliquots from the same samples. So all the samples will be directly comparable across the different platforms. And again, the performance of the platforms will be evaluated based on the results for these reference samples. When submitting the data, process data should be provided as a single table with a column for each test sample and a row for each gene or measurement. Values in that table should represent a measure of absolute or relative expression of each feature and is intended for differential expression and signal-to-noise type analyses. Solvers must provide a clear and unambiguous mapping from the columns they provide to the sample IDs and from the rows to the gene IDs in their transcriptome annotation for each species. And the solvers should be willing to furnish the raw data upon the request of the sponsors. Data from the reference samples need to be submitted by June 14th, 2021. To be considered for the award, you need to submit not only the process data file for the reference samples, but also annotation files that will help determine the overall coverage of the platform and how well the genes can be mapped to potential pathways and used for inference of mode of action. Finally, there will be a technology description document where you will lay out the title of your platform, a brief description of your assay platform, including the conceptual and theoretical basis of the assay approach, focusing on details that are unique to your method, specifying any quality control features of your platforms, the sample requirement in terms of the types of samples, the mass of RNA needed, and so on. Per sample cost of the analysis should be estimated. Any downstream data analysis requirements that are needed. So for example, if proprietary software may be needed to process the data, that should be clearly indicated. And finally, a path to the commercial viability, how you would bring the platform or technology to market in a way that could meet the demands of these emerging high throughput screening programs needs to be laid out in that technology description. Once you've submitted this information, the submissions will be judged based on 3 overall criteria. First, the quality and performance of the platform and the quality of the associated data. Second, the economic and commercial viability of the platform and how clearly a pathway to commercialization or the ability to meet the demands of these high throughput programs, how well that's laid out. Third, the extent of the transcriptome coverage, what proportion of the overall transcriptome of these organisms is covered by the technology. A detailed breakdown of these scoring and evaluation criteria can be found at the website with the challenge rules. And the overall award will be announced somewhere around the end of September of 2021. So again, if you think you've got a solution, if you think you have a technology that will work for this particular application, we encourage you to participate. Why should you take the time and effort to participate? Well, again, there's opportunity to win a $300,000 cash award. There's also opportunity to demonstrate the capabilities of your technology and organization to a group of government, industry, and academic organizations with a strong interest in using those technologies. Positions your organization to serve this growth market as these high throughput assays are brought online, and also brings attention and awareness to your products, showing that you were competing in this challenge and basically showing how you were able to respond to this need. It's also useful to participate because cool science can emerge from this. So for example, improving the understanding of the molecular physiology of non-mammalian organisms, as we're able to test the response of these organisms to tens, hundreds, potentially thousands of different types of chemical stressors. We can start to see how some of the less understood parts of the genome of these organisms is responding, and from that start to infer what kinds of functions some of those unannotated genes, some of those unknown pathways might have in these organisms. Can also contribute to the maturation of transcriptomics as an accepted tool for risk assessment and regulatory decision making. And finally, contributing to this challenge can help protect the environment by providing us with the low-cost tools and technologies that we need to generate more chemical safety data and make sure that we're covering the breadth of species that we're charged with protecting. So again, thank you for tuning in to the webinar today. Here's the link to the challenge website. I encourage you to go and take a look. Additional details, all the rules, the documentation that you'll need are there on the website. And at this point, we'd be happy to answer questions. Thank you for participating, and we hope to be talking to you in March.